Hey everyone, welcome back to the second session of the Seed Biology and Evolutionary Ecology talks this morning. We're going to leap into 6 more of our 8-minute talks and then a couple shorter ones, and then our speakers will join us for a Q&A at the end. So remember, questions into the live Q&A box if you can make it work, and if not, just put them into the discussion forum as we go. Over to you, Kali.
Hi everyone. Today I'll be talking to you about predicting germination sensitivity of a number of species from threatened ecological communities in northwest New South Wales. The Brigalow Belt South bioregion predominantly occurs in southeastern Queensland, but around 1/5 of it extends into northwest New South Wales. It's characterised by hot summers, cool to mild winters, highly variable annual rainfall, and some areas that are classified as semi-arid. At least 60% of the region has been cleared for cropping and grazing purposes, and less than 3% of the region is under any conservation mechanism. So consequently, there are several threatened ecological communities within the region. Brigalow, semi-evergreen vine thicket, and Cadelia pentastylus scrub are some of the most notable communities in the region. There are many knowledge gaps that remain surrounding the community and the species within. Notably, seed germination requirements and tolerances of many species are unknown. Now, climate change is a threatening process for the region and the communities within. The future predictions include an overall temperature increase of between around 0.5 degree to 1.5 degrees Celsius, lower overall spring and winter rainfall, and an overall reduction in soil moisture due to that reduced rainfall, as well as the prolonged onset and frequency of droughts. So being able to explore the width of the temperature and moisture niche of species helps us predict germination in situ and identify those species that may be highly sensitive to climate change. We run species through a workflow in the laboratory to test their temperature and moisture tolerances. For temperature, we use a thermogradient plate, which allows us to look at a multitude of alternating and constant temperatures between about 5 to 40 degrees Celsius. And this gives us a lot of germination data to play with using fewer seeds than what we would via traditional means by using multiple incubators. To analyze this raw data, we've developed a statistical framework which allows us to predict germination at the local site for that species under current and future climate predictions. I'm not going to go into the details of this statistical framework, but encourage you all to listen up to Justin Collette, who will give a presentation later on today about the methodology and go into the details of the statistical framework. To investigate the moisture tolerance of species, we use polyethylene glycol, or PEG 6000. PEG is a water-binding polymer that's not absorbed by the seeds. It binds to water molecules and prevents that water from entering the seed cells. So this allows us to induce water stress reproducibly in the lab. We use a number of treatments for testing moisture stress, including a control, a low, a medium, and a high treatment. And we place seeds in an incubator to germinate at optimal temperature, which we determine by our thermogradient plate results. Now, here are some of our results so far. There are 4 species on this slide, which are co-dominant canopy species from the Brigalow threatened ecological community. And if you look at the rasterized quilt plots along the top row, these are our raw data results from our thermogradient plate experiment. So you can see that each species has quite a wide temperature tolerance and quite high overall germination across all the temperature combinations. Now, we use this raw data to run through our statistical model to get a predicted set of data so we can look at germination across all temperature combinations. And you can see, if you compare the top row with the middle row, there's good overall fit, good synergy between our raw and predicted data. We then take this predicted data and model it with climate to look at germination at the local site under current and future climate. And you can see for all species, there's very high, very consistent germination overall in both the current and future predicted climates, with very little change between the two. And here are the moisture stress results for 3 of these species. Each species is represented by 2 graphs, the one on the left for final proportion germination and the one on the right for time to 50% germination. In the case of the 2 eucalyptus species, there is good germination at both the control and low moisture stress treatments, while there's a reduction, but some seeds are able to germinate at medium moisture stress levels. The acacia, however, shows really good germination at control and low moisture stress treatments. And while there's a reduction in germination in the subsequent treatments, there's still seeds that are able to germinate under these medium and high moisture stress treatments. Now, here's where it gets a little bit interesting when we look at the 4 dry rainforest species that we've run on the thermogradient plate so far. If I draw your attention to the rasterized quilt plots along the top row, each of these 4 species show a more narrow temperature tolerance for germination compared to the 4 woodland species I've shown you previously. When we run these data through our statistical framework, each species shows a high correlation and good fit for our predicted data. Now, if we focus on the lower row where we look at predicted germination at the local site under current and future climate, all 4 species currently show maximum germination at warmer months during spring and summer. But in the future, there's a seasonality shift for each of these species away from these warmer months to higher germination overall during the cooler winter months. The moisture stress results of 3 of these species are also quite interesting. Not only do they show a narrow temperature tolerance for germination, but also a highly sensitive response to any level of moisture stress. 2 species are able to have some germination under the low moisture stress treatment, while in the case of the Electrion, we're seeing effectively 0 germination at any treatment that has a level of moisture stress to it. So these are some really cool early results from our work, and we're seeing different germination strategies of species tested that reflects their habitat. Seeds of woodland species show a broad temperature range for germination, as well as little change in predicted germination under future climate. The seeds are also able to germinate at medium and high moisture stress levels, so these species may be more likely to withstand future climate pressures. By comparison, dry rainforest species that we've tested are showing narrow temperature ranges for germination, which is then reflected as a seasonal shift for germination in the future away from the summer months to the cooler winter months. The seeds are also highly sensitive to any level of moisture stress, so we may need to flag these species for future management actions. If we think about the predicted future climate for the region where there's reduced spring and winter rainfall, this could have dire consequences for these species that are showing a preference temperature-wise for these cooler months. So this is a really neat approach for examining the temperature and moisture effects on germination, which then enables us to try and predict the impact of climate change. Ideally, we want to back up these experimental trials in the lab with field trials. But this work helps us to prioritize species for conservation management. It provides important information to the restoration industry for helping with their direct seeding efforts, and also assists with developing propagation protocols in nurseries. So this work is currently ongoing, and we're hoping to do further investigations of additional species running through this workflow, as well as looking at variation among years and different provenances. And I'd just like to thank everyone who's been involved in this project to date. Thank you.
Hi everyone, my name is Gemma Hoyle and I work at the National Seed Bank at the Australian National Botanic Gardens. My talk today is on the impact of light on germination in Australia's tropical mountain cloud forest. This work is thanks to funding from the Ian Potter Foundation and the Wet Tropics Management Authority. Tropical mountain cloud forests are defined by high altitude rainforest and frequent or persistent ground level cloud. Globally, cloud forests are critically threatened by climate change. Here in Australia, forests like these are restricted to the Wet Tropics World Heritage Area of Northeast Queensland. The areas in red here are above 1,000 metres and are where we do our seed collecting. These forests are home to over 70 plant species that are not found anywhere else in the world. Modelling studies have predicted that suitable habitat for more than half of these species will decline or be eliminated altogether by 2085. Although these predictions are alarming, they assume that the current niche occupied by each species is essential for its survival, when in fact virtually nothing is known about factors that determine the regeneration and distribution of these species. An important factor for seed germination is light. light is particularly interesting in a cloud forest because canopy cover, leaf litter, and cloud immersion all affect the ratio of red to far red light that plants are exposed to. So unfiltered sunlight is equal parts red and far red light, pretty much. The red to far red ratio is 1.2. As light passes through a leaf canopy, red light is absorbed, so the red to far red ratio decreases. As light passes through a secondary or under canopy, more red light is absorbed and the ratio is even lower. Beneath the leaf litter, on the ground, the ratio can be as low as 0.1. So this reduced red light is generally understood to inhibit seed germination. That said, the ratio beneath the gap in the canopy can be as high as unfiltered light. So this red to far red ratio has been shown to influence seed germination of many temperate species and some tropical rainforest seeds. We asked in this study, do cloud forest seeds require light for germination? And do they differentiate between different red to far red ratios? We investigated these questions on germination of 6 previously unstudied species, including endemic and more widespread herbs, shrubs, and trees. We created a gradient of red to far red equivalent to those that seeds in situ would experience using natural sunlight and polyester filters. So this figure shows the mean red to far red ratio created by the different filters. And we sowed seeds onto agar, placed them underneath the filters. We also wrapped seeds in alfoil to create a dark treatment here. The glass house averaged at about 25 degrees C, and then we calculated final germination after 10 weeks. These figures show the light gradient on the x-axis and final % germination on the y-axis. Germination of Dracophyllum sayeri was highest in unfiltered light and decreased with decreasing red light down to low germination in the dark. Results suggest that this species is more likely to germinate beneath a gap in the canopy or on a rocky outcrop, and less likely to germinate beneath a leaf litter or a thick canopy. And in fact, in situ, this endemic tree is usually found growing in open sun or exposed places. One disadvantage to germinating in full sun, however, is that seedlings are then usually prone to desiccation stress. However, Dracophyllum seedlings are known to have tough, drought-tolerant leaves, which may explain why it's able to establish in full sun. In contrast, Lewandrowski australiana, this one displayed an absolute light requirement for germination, so no germination in the dark, and germinated well across the entire gradient of red to far red ratios. It didn't germinate very well following 10 weeks of darkness when these seeds were unwrapped and placed into unfiltered light. So results suggest that germination, that this species in situ will germinate regardless of light quality, but seeds won't germinate in the dark, and that they might become incorporated into the soil seed bank. Lastly, Garnia siberiana. This one germinated better beneath most of the filters and in the dark than it did in unfiltered light. In situ, this widespread herb is a very dense plant, therefore dispersed seed on the ground would have very little light. It clearly has a preference for germination beneath the canopy or leaf litter, or maybe even from the soil seed bank. Perhaps germination in the shade is a seedling survival advantage. So light would appear to play an important role in the timing and placement of germination in the wet tropics. Even in this small data set, we see variation in germination response among seedlings. Some species were capable of germinating under a wide range of light quality, while for other species, the red far red ratio appears to permit fine scale discrimination of light conditions for germination. Following on from this work, we would like to explore whether seed mass, plant distribution, growth form, seed dormancy, and other traits are associated with response to light. We'd like to look at how subsequent seedling establishment is impacted by light quality, and how predicted changes in canopy density and cloud immersion in the future might affect regeneration from seed in this part of the world. If you have any insight to share on this study or its findings, it would be great to hear from you. And thank you to my co-authors and other project colleagues, and thanks to the conference committee for having me.
And today my presentation will be on seed traits from banded limestone formation species in semi-arid ecosystems. Banded formations are characteristically rich in iron. They primarily, most of them occur about 450 kilometers east and northeast of Perth in the goldfields and into the Midwest region. We're looking at very unpredictable rainfall events, very hot summers of about 40 degrees and very cold winters, sometimes reaching 0 degrees. At the same time, these ranges are very often mined for their resources. They're extremely species rich. You'll find a lot of endemic species in these areas, and at the same time, also home to a number of priority and threatened species. So when looking at this rock, it actually looks quite a bit of a hostile environment. This rock can heat up to about 75 degrees in summer quite easily, but there's signs that this rock can actually hold on to life. So we can see plant life, we can see lichen on the rock. When looking into shallow salt pockets, as we see in this center picture, you can actually see litter depositing onto the soil layers. This will help reduce evaporative water loss. And then further looking at 20 centimeters and 50 centimeters sub-rock. So this was us opportunistically, before this area got mined, taking some soil samples from these rocky sites. We actually found moisture available at about 35% gravimetric weight. So for this soil type, we're actually sitting at about 75% from fuel capacity. So in other words, dare I say, the rock is almost acting like a sponge and providing resources to sustain life. So just acknowledging some of the work to really help paint the picture here. In the top left, we in previous studies have identified that for a lot of our threatened and rare species that have very slow germination rates, their recruitment events are really driven by these 1 in 30 and 1 in 60 year rainfall. So these really rare rainfall events. And then just acknowledging Roberta Darrell's and Fernando Silveira's work from Brazil. It's all about the microsites. So location, location, location. More importantly, seed functional traits can really help us understand species requirements in order to get them back in restoration sites, but also from a single species perspective for a lot of our rare and threatened species. So for this little guy, just synthesizing the information, it's all location, location, location. And it's all about understanding the micro niche. A lot of our BIF species, unfortunately, there's a massive gap. We don't know what the moisture and temperature requirements are. So I'm really looking forward to the work coming up by Shane Turn and also by Lucy Commander, who's also here at this conference, just highlighting some of those dynamics. So here's one of our unusual rare suspects. This is Tetratheca arabescens. We've been working on the species for the last 4 years in a translocation program. He occurs only across 3.5 hectares. It's quite a narrow range. In a previous seed collection prior to this, there was no germination at all from fresh seeds. So in this collection here, we can really see when it shows the variability in collections, number one, but number two, when we address dormancy, so this is a hydrothermal time experiment that we ran. So this combining different temperatures with different moisture combinations, really identifying what the thresholds are that limit germination. When we address dormancy, we can actually improve germination across different temperature regimes, but also into water stress. So we're looking at this -1 megapascals and at 15 degrees. Some other cool results relating to this guy. At any alternating temperatures that we tested, germination could not occur. So we believe that alternating temperatures are reminiscent of seeds being very close to the soil surface, and this might be a very risky environment for the species. Some other data here for Banksia borea, it's another narrow range endemic, just really showcasing that genetics really matters. So these are all genetically different populations. They're also slightly climatically different. So the Kuli-Nobbing population is the one that has slightly higher evaporation rates. But it really shows that, again, under these stressful conditions, we're getting germination from the Kuli-Nobbing banded ironstone range compared to the other ranges. So just going into some of the community dynamics here, and some really cool trends are that some of the species, some of the more generalist species are actually showing quite wide temperature responses. These guys quite commonly occur throughout BIF, but also on the slopes and the flats. I also included an invasive weed, which is this ruby dock over here. The other species that we're looking at, so our narrow range endemics, the Tetratheca arabescens, and also that Banksia borean, and a few of the grasses were also showing quite narrow, so winter kind of conditions for germination. From the moisture perspective, some really cool results again. So we're looking at some species that are really sensitive to moisture. So at 0, we're looking at free available water, and at -1.5, we're looking into conditions almost at drought. So for other species, some can actually push that envelope and we're still germinating under those drought conditions. Furthermore, just summarizing those results together, we generally found differences between short range and also those generalist species. So our generalist species are germinating much quicker, and that's represented in shorter thermal time and short hydrothermal time components. Our generalists were also showing much wider thermal ranges compared to our short range endemics. And one of the traits that I thought was really interesting was the maximum germination temperature and how generalists and short range endemics were showing distinctly different regressions against seed mass. So we ran a few other hydrothermal experiments, just trying to understand some common patterns. And these were the two main patterns that we were getting. So winter germinators like this Tetratheca arabescens, and this was modeled for 60 days. And for this species, despite having narrow temperature conditions, it was actually able to germinate to a much wider water stress compared to the Acacia tetragonophylla. So that's our generalist species, a really wide temperature envelope there, but slightly more narrowing water stress conditions. So just in closing, where we'd like to go in future, we previously have published an edaphically informed species distribution. Also, we've taken edaphic factors like slope, clay, sand, and silt content to help us predict where species could further occur beyond their own niche, or where we've mapped them to occur. The next whole idea is to actually combine these hydrothermal time models in with these edaphically informed models to furthermore predict where we could potentially identify germination features in the environment. So with that, just thanks to everyone, my co-authors, and also to our army of volunteers. And please get in touch if you have any questions. Thanks.
Good morning, everyone. I'm Rocco Notarnocola, and today I'm going to talk about life history responses in seed traits of the alpine herb Wallenbergia cerasea to warming temperatures. Seed size and seed number are both adaptive life history strategies. In fact, investment in seed size, producing larger seeds, usually corresponds to higher provisioning to the seeds, increasing survival and competitiveness of number increases the chances that more seedlings will be successful and establish. However, since resources to invest in reproduction are limited, these two life history strategies are assumed to trade off. Therefore, a change in one trait should correspond to a change in the other in the opposite direction. So far, mixed evidence of this trade-off were found in plant species at the intraspecific level. Global climate warming is threatening biodiversity all over the world, and climate models project rising temperatures up to 5 degrees by 2100 under the worst-case scenario. There is evidence that warmer growth temperatures will induce plastic responses in plants, and therefore rising temperatures will likely induce responses in seed life history traits too. So if plants, for example, invest more in seed size, producing larger seeds, then under the assumption of a seed size number trade-off, a concurrent reduction in seed number should be expected. While if both responses are in the same direction, then no trade-off is detected. Few studies so far investigated the effect of growth conditions on this trade-off, but this is important because these responses might in turn affect germination responses and on a larger scale population dynamics. Why alpine plants? We think that alpine environments are a good system to investigate the effect of warming on responses in seed life history traits, because temperatures have risen twice as fast in alpine than lowland environments. In addition, alpine plants possess complex germination strategies, such as staggered germination, where in the same batch, different types of seeds are produced. Some germinate soon after dispersal, and they are non-dormant, while others germinate only after snow melt, or even later, and these are dormant seeds. For this study, we used Wallenbergia cerasea, which is an Australian endemic herb with staggered germination, and evidence of plastic responses to warm growth temperatures were found in these species. So the questions we're trying to answer in this talk are whether there is evidence of a seed size number trade-off in these species, whether warming affect this trade-off in using plastic responses in seed life history traits, and whether that in turn affect fitness. So to investigate plastic responses to warmer growth temperatures, we grew parental individuals from 30 families in growth chambers under simulated cool and warm climate with relevant changes to temperatures and photoperiod every 15 days. And we grew these plants for their whole lifetime for about 200 days. And we measured their lifetime flower production and collected all the capsules they produced. From these capsules, we obtained the seeds that we use to measure seed size as the average weight of an individual seed and seed number as the total seed production. And we used these seeds in germination assays where we evaluated total germination and seed viability. Moving on to the results, we found only weak evidence of a seed size number trade-off in Wallenbergia cerasea, and the slope of the reaction was significant only under cool growth temperatures, while the trending positive regression was under warmer growth temperatures was not significant, nor was the interaction. Warmer growth temperatures induced investment in flowers, as you can see in this graph that presents a reaction norm that is usually used to show plastic responses to growth conditions. And this investment in flowers suggests an investment in the seed number life history strategy. And we concurrently observed a reduction in seed size, somewhat confirming the assumption behind the seed size number trade-off. However, investment in flowers was not successful, as many less seeds were produced under warmer growth temperatures, because under these conditions disrupted flower and seed development, as many flowers were aborted and many seeds were empty or immature. The concurrent reduction in both seed traits resulted in a positive trend, although not significant, in the slope of the regression. We also found that seed size was positively correlated to seed viability, and this correlation was stronger under warmer growth temperatures, suggesting that the smaller seeds produced under these conditions were also less viable. And this is further evidence that investment in flowers was not successful. As a summary of our results, we calculated a fitness index as the product between seed production and total germination. And as you can see here, warmer growth temperatures severely reduced parental success. So in summary, the weak seed size number trade-off in Wallenbergia cerasea was alleviated under warmer growth temperatures as both seed traits were reduced. Under warmer growth temperatures, plants did invest in the seed number life history strategy, producing more flowers, but this was unsuccessful under our experimental conditions. And it would be interesting now to test these results under natural conditions. Overall, parental fitness was severely reduced under warmer growth temperatures, suggesting that alpine plants might soon be under threat due to climate warming. So future steps, we are now evaluating whether these responses in seed life history traits affect germination strategies, i.e. dormancy levels. And with that, I would like to thank all the people involved in this study, the ANU Plant Service team, and all the past and present members of the Nicotras and Krug's lab. Thanks for your attention.
Hi everyone, today I'm going to talk to you about polyploidy and seed germination. My name's Lydia and I'm from the Centre for Australian National Biodiversity Research, which is a joint venture between CSIRO and the Australian National Botanic Gardens. So I'm actually going to start at the end, start at the end with the key points that I'd like you to take away from this presentation. And so that is that polyploidy affects germination. This happens at multiple levels within and among species, and we need to identify its presence for conservation management. And we still have work to do on understanding the ecological consequences of polyploidy. So how did I arrive at these conclusions? First, I'll start by just a quick introduction of what polyploidy is. So it's a heritable condition of possessing more than 2 chromosome sets. And it's something that's rare in higher animals, but happens quite frequently in plants. And it's occurred repeatedly over the evolutionary history of flowering plants and is often linked to speciation. So why does polyploidy matter? Well, in conservation management, we sometimes find ourselves in a situation like this example of a sedge in Western Australia, which was prioritised for mine site restoration, but occurs as small fragmented populations. And in this example, we found that populations tended to be mostly one ploidy race, but there were a couple naturally occurring where they where ploidy was mixed, but generally populations tended to be one ploidy level. And so this helps inform what we might use for mine site restoration, and we need to be aware of what's being mixed in those restored sites, because if different ploidy numbers are mixed, so for example, if diploid plants and tetraploid plants are co-occurring, they can produce triploid offspring, which actually uneven ploidy numbers tend to often produce to be sterile or to produce sterile seed if they do produce seed. So this is not something we would want in a restoration context. It would make it quite challenging to produce a long-term sustainable population, but it is something that we take advantage of in everyday life. So for example, most cultivated bananas are triploids and sterile and therefore seedless. So there can be sometimes benefits to polyploidy, and certainly ecologically, polyploids or plants with more genetic material tend to be larger. They've been found to be more stress tolerant. They're known to sometimes be more invasive and to have greater reproductive output than diploids. So this matters ecologically as well as in conservation management. And it's interesting working on polyploidy because it's not something that you can easily tell from looking at a plant. So they're usually morphologically cryptic, and it requires a fair bit of work back at the lab to identify the ploidy status of a individual. But more recently, that's become quite cheap and rapid to do using flow cytometry techniques. So this has really opened up the ability for us to look at things like polyploidy in seed germination studies. So the first example that I will share is an example looking at within species variation in polyploidy. So this is kangaroo grass, which occurs across much of Australia and is known to have diploid, tetraploid and other ploidy races. And colleagues published in 2017 a paper where they took diploid and polyploid plants from diploid and tetraploid plants from 3 different sites where they co-occur, and grew them together in a common garden experiment and exposed them to drought and heat stress. And the really interesting result of that was that the tetraploid plants were able to produce 4 times the seed of the diploids, even under those drought and heat stress conditions. So we were really interested in understanding the performance of those seeds and their ability to germinate. And I don't really have time to go through too much detail of the results, but it can be summed up in showing a few of the highlights, and that is that the polyploids produced much larger seeds than diploids. The polyploids also tended to have a deeper dormancy than diploids at dispersal. And once that dormancy was alleviated, we found that germination was lower in polyploids than diploids, and that continued under stressful conditions, for example, water stress that you see in the figure here. but overall the polyploid seedlings were larger than diploids. So taken together, all of this told us that polyploids are less likely to germinate, but more likely to survive than the diploids. So now I'm going to change completely from that example where you saw variation within species and really big differences among populations. This time we're going to look at variation among species or among taxa. So this example comes from the family Rhamnaceae, the genus Pomaderus, a genus of shrubs. And we were interested in this group because there are so many listed threatened species. So you can see the little red asterisks on the figure there. Almost or something like 2/3 of the taxa from this study are listed as threatened. And we use flow cytometry to look at the to identify polyploidy in this genus, and we found that it was very widespread across the genus, and you can see there that almost half of the species were polyploids. So following on from that, we looked at the germination ability of the seeds, their dormancy breaking thresholds in response to heat, and also the seedling growth of species. And what we found was that a few of the headline results at least was that the polyploids had larger seeds than diploids. They had much faster germination, so half of the seeds germinated in 20-something days rather than 40-something days. And the polyploids had much larger seedlings than diploids. So in this example, the polyploids were larger and may have a competitive advantage over diploid taxa. So bringing together the results from these studies, we can see just from these few either species or groups of taxa where we've looked that polyploidy does affect seed germination and creates quite big significant differences in germination, and this is at multiple levels, either within or among species. And so I think moving on in the future, we really need to look more closely at identifying the presence of polyploidy for conservation management. And we also need to move beyond the seed and seedling stage to better understand the ecological consequences of polyploidy. So just to end, I'd like to acknowledge the traditional owners of the lands where this research was conducted, and also all of my co-authors for the abstract and the presentation, and in particular Millie, Jason and Stephanie, whose student projects form a lot of the research that's been shared here. And finally, the National Seed Bank team for their feedback and good comments on a draft version of the presentation. Thank you.
Thank you. Good afternoon, everybody. And today I'd like to talk about some research work that was undertaken by a student at Massey University, a master's student who undertook some research in looking at desiccation tolerance in 2 New Zealand Fabaceae species. So that student was Yukai, co-supervised by myself, Marion Mackay, and Jayanthi Natarajan. But first, a little bit of background on the 2 species. The Cleanthus genera has 2 species within New Zealand, both of which are endemic, Cleanthus punicis and Cleanthus maximus. The common name for the flower is, as the plant, is kakabeak. That's because, as you can see from the flowers, the shape of the flower is similar to the kaka bird, which is a bird that's endemic to New Zealand. Both these species are considered to be nationally threatened. Their situation is critical. In the wild, there is only one species, one plant of Cleanthus puniceus, and for Cleanthus maximus, there is less than 200 plants still in the wild. So they are considered to be nationally critical, and therefore we have a strong interest in conserving the seed within a seed bank. The other species that I'm going to look at is Karmachelia muritai. So Karmachelia are a group of 24 species, 23 of which are endemic to New Zealand, and 1 is found on Lord Howe Island. Again, this plant is nationally endangered and is therefore also a candidate for conservation within the seed bank. Both genera are within the Fabaceae, so as you would expect, they have hard seed that is water impermeable. So in looking at trying to confirm that both these species, these species within these two genera were desiccation tolerant, we made an observation, and first I'd like to look at that of Cleanthus Provenances and Cleanthus maximus. So we took seed from 4 different locations, two provinces of each, and they were desiccated in silica gel until they reached target moisture contents of 5%, 2.5%. And then we assessed moisture at the end of that desiccation time using the forced air oven, 103 degrees for 17 hours. Seeds were then placed on for germination without scarification. They were allowed to imbibe, and after 21 days, if for any seeds that had not germinated, they were scarified to enable germination to proceed and to check that the seeds were viable. As a result of the desiccation, you can see that if you look at the Cleanthus maximus here, you can see that it's at 7.4% moisture. There was a reasonably high level of germination compared with the other 3 provenances that were studied. But what we discovered when we were drying the seeds, desiccated the seeds down further, was that at 5.9% moisture, in fact, there was a high level of germination even without scarification in all 4 provenances, and similarly at 2.5%. So clearly, with drying to ultra dry moisture, these seeds were losing water impermeability. The seed coat was losing water impermeability. To study this further, we looked at the structure of the seed coat and using 2 approaches, one staining with methylene blue and one looking at scanning electron microscopy. So just to explain the areas that we're looking at, this is a typical Cleanthus seed. So what we're looking at is the micropyle and hilum area, the lens area, and what we call the extra hilum area, which is essentially the seed coat outside any of those areas of the lens, the hilum or the micropyle. What we did was we imbibed the seed, and in this case, I'm only presenting because of time, I'm only presenting the data for Cleanthus maximus seed. So we immersed the Cleanthus maximus seed in water for 24 hours, and we then followed that by uptake of 1% solution of methylene blue for 24 hours. Various areas of the seed coat were obstructed with Vaseline, which was used as a blockage for water going in. And we looked at the movement of methylene blue into the seeds at the 2 different target moisture contents, 2.5% target moisture content and 5% target moisture content. As you can see, that in the absence of Vaseline, methylene blue was able to enter the seed from all locations, from the extra hilum area, the hilum area, and the lens area. Similarly, when we blocked the lens area, we can see that methylene blue was able to enter the seed through the extra hilum and hilum area. And similarly, when we blocked the extra hilum area, the lens and the hilum area, methylene blue was able to enter through the extra hilum or general seed coat. What we then did was we looked at where the impact of the desiccation to 2.5% target moisture, 5% target moisture, and the initial seed moisture content on the structure of the seed coat using scanning electron microscopy. And what we can see is that cracking was observed to both the extra hilum area and the lens area of the seed coat at both target moistures, 2.5% target moisture content and 5% target moisture content. But there was no cracking observed at the initial moisture contents. Interestingly too, at 2.5% target moisture content, there was cracking to the hilum, but not at 5% target moisture content. This data can contrast with some earlier work that was done by another PhD student, Park Myung-Ju, in which when Park Myung-Ju desiccated both species of Cleanthus to 2.5% target moisture content, she did not find any cracking to the hilum, only to the extra hilum and lens areas. Similarly, some work by Park Mianju with another species of Carmichaelia wilsonii, when desiccated to 5 and 3.7% moisture content, there was no cracking to the seed coat observed. However, in contrast, you can see from this data here that when Carmichaelia muritai was desiccated over silica gel, there was cracking, there was, loss of germination in terms, loss of hard seed. Interestingly, at 7.8% moisture, there is already a percentage of hard seed, but as the seed is desiccated to 5.9 and 2.8% moisture, hard seededness is lost. So again, we stain the sections, the seeds, with methylene blue after first being imbibed in water for 24 hours at 2.5% and 5% target moisture contents. There was staining of methylene blue in the absence of with no Vaseline. Also, with Vaseline to the extra hilar and hilum area, there was still staining in the lens area. You can just see that in the diagram, suggesting that the methylene blue was able to enter through the lens. And again, if we look at the edges of the pictures on the far right, where the Vaseline was applied to the lens and hilum area, you can see that with those areas being obstructed in terms of methylene blue entry, there was entry of methylene blue through the seed coat, through the extra hilar area. However, in contrast to the Cleanthus species, there was no correlation between methylene blue and what we're observing in terms of the seed coat surfaces of the Carmichaelia muritai. So if we look at this series of scanning electron microscopy graphs again, you can see that there was cracking to the areas out near and surrounding the hilum and micropyle, but no cracking to the extra hilar or lens area in any of the target moisture contents, nor the initial moisture, nor seed at the initial moisture content. And so in summary, seeds at 2.5% moisture showed cracks in the hilum and extra hilar area, but it was only cracks were only observed in the extra hilar area at the target moisture content of 5%. So this may suggest that different areas of the seed coat are responding to the drying regime in terms of different cracking. So clearly it's suggesting that the hilum may be more resistant to cracking than the other areas in the seed coat. And as I said, that for seeds at a target moisture of 2.5 and 5%, Carmichaelia muritai had cracks in the hilum and micropyle area, but no cracking was observed in any other areas of the seed coat. And this was a particularly interesting and confounding result, in so much as, despite there being entry of methylene blue through all areas of the seed coat, that wasn't shown as a correlation to cracking damage. So it's still not clear what the reason for that is, and also that there was no cracking was observed at the initial moisture content, despite 50% of the seed being water impermeable. So this raises some questions in terms of our long-term conservation of these species. There looks to be differences across provenances. So, for example, the data that Mianju Park found in her study differed for Cleanthus and both Cleanthus species to what Yukai was finding in her study. And there may also be species effects here. For example, comparing Carmichaelia muritai and Carmichaelia williamsii. This may also possibly reflect, given that we know that hard seededness develops during the seed development, the later stages of seed development. This may also reflect different developmental environments of the seed that we studied. So clearly there needs to be more work in terms of looking at a wider range of provenance for this. The other question is that if we are losing the integrity of the seed coat at very low moisture contents, what implications does that have for long-term storage of these seed at very low seed moistures? Remembering we want to be able to store these seed long-term, because all three of the species that I've talked about are threatened, seriously threatened within the wild. So in closing, I'd like to acknowledge the Manawatu Microscopy and Imaging Centre at Massey University for help with the scanning electron microscopy, Jessica Snell, who previously worked in the Sea Bank at Massey University, and the staff of Otari Botanic Gardens for helping UKI with this seed collection. Thank you for your attention.
Hello everyone, I am Widjaya from Research Center for Plant Conservation and Botany Gardens, Germination Institute of Sciences, Seed Bank Bogor Botany Gardens. Sampole has been cultivated in West Java and has become a local fruit planted in the gardens or yards. Sampole, the germination could be inhibited by their hard seed coats related to their impermeability. Question and method. Harvesting fruit, characterization of fruit and seed, extraction fruit, measure of moisture content, torsion seed weight, seed classification, seed storage, seed germination. Seed germination was not significantly different at different weight and storage periods. The coefficient of germination rate of sample seeds were significantly different at all shelf lives. Sample seeds with shelf life of 4 and 6 weeks germinated faster than those stored for 0 and 2 weeks. Simultaneous germination coefficient of sample seed was significantly different from the shelf life of 6 weeks, but not significantly different at shelf life of 0, 2, and 4 weeks, as well as for various seed weight. There is no interaction between egg and weight shifting sample seed germination of seed with a total of 92 to 97%. Seed storage shelf life significantly accelerates seed germination time with germination of up to 60%, first and ideal last day of germination, while the seed weight has no significant effect. The last day of germination was significantly affected by seed storage, but not influenced by seed weight. This ends my presentation. As welcome for any comment and question. Thank you.
Thanks for joining our presentation, Soil Seed Bank Diversity under Variation in Fire Regimes in a Temperate Heathland. This research was conducted on Gundishmara, Japarang, Orchabaluk, Yadwa, Yadwajali, Wagai, and Jubagul country. Heathlands and species in Garryward are species-rich and fire-prone. They have fire-related traits, such as fire-stimulated seed release, germination, and resprouting. But anthropogenic changes to fire regimes threaten plant species. Fires that are too frequent, infrequent, or severe can harm plant populations. Therefore, we want to understand how fire regimes, particularly fire interval and fire severity, influence species richness and relative abundance in the Garryward Heath ecosystem. Fire regime includes temporal, spatial, and intensity patterns of fire. Most studies that quantify fire effects use one measure of fire regime, typically time since fire. However, we use time since fire, fire interval, and fire severity in combination. Furthermore, conventionally, plant diversity is quantified above ground only. We use the soil seed bank study in combination with the extant vegetation, as they're both important for post-fire recovery. We selected 57 sites from Garryward in southwest Victoria with varied fire interval, time since fire, and fire severity. This map demonstrates the number of fires at each site from 1 to 8 since 1939. This map shows a small sample of sites and how time since fire has varied among them. Finally, this map shows 2 sites on a 2006 fire severity map made by the methods of Collins et al. 2020. One site is high severity on the lighter colour and one is low severity on the darker colour. At each site, we collected 81 soil samples. Samples were air dried, sieved, and divided into 3 treatment groups. One treatment was a 90-degree treatment with smoke, one 60-degree treatment with smoke, and one group was left untreated. Samples were then moved to an irrigated polyhouse alongside 10 control trays. 14 weeks into the study, and species are being recorded once identified. Some species that have been identified include Hydrocotyle laxiflora, Canetia prostrata, and more. For the first 5 weeks, we used toothpicks to record weekly germination and species. This graph shows the percentage of types of germinants for the first 5 weeks to the finest possible identification at that stage. The first few weeks were dominated by grasses across all treatments, with Asteraceae also common in the low heat treatment. Other types of plants started to emerge in later weeks. The next steps will be to record seedlings until April 2022 or until seedlings cease to emerge. Use regression analyses to explore measures of plant occurrence and species richness in the soil seed bank as a function of fire history and compare those relationships to the extant vegetation. Thanks for watching our presentation. You can find out more about this work by emailing me, connecting me on Twitter, or checking out the website.
Thanks everyone for a fabulous set of talks in that session. There's so much ground we've covered. I might start by drawing your attention that there are many more questions in our question box than we're going to be able to answer. So I'm going to try and ask if we can get through them. And I'll start with a question from Jose for Nathan, I believe. If I can find it now, it's gone. Ah, there we go. Jose raises the interesting point that there's a difference between germination and establishment. So Nathan, can you chat a little bit more on how we get around that, given our experimental logistical constraints?
Yeah, thanks for the question. I guess it's important to mention that the methodology that we're using for the thermogradient plate and the PEG trials are a first step to explore and prioritize species and guide us on the next step. And the next step is to conduct field trials and verify what we're seeing in the lab is what we're getting out in the field. PEG is a uniform medium and soil very much isn't. And there's some very recent research that has shown that the results you get for PEG in terms of germination doesn't match what you get in some soils. So yeah, we really need to be able to use the data that we get from the lab to then conduct field trials in the seasons where we expect germination to be high, and monitor the soil condition and the moisture to make some comparisons between the lab and the field.
Thanks, Nathan. I might move now to a question for Wolfgang from Marlene. And she's interested in a question about follow-up. So it's kind of similar to what we've just given to Nathan. It's the what next kind of question, right? Have you thought any further about impacts on the seedlings once they've germinated?
Absolutely. Thank you for your question. That is a really great question. The challenge that we've got working in these rocky environments is really understanding those unseen processes to the eye. So how, after seeds have germinated, how do the seedlings grow their roots down through those cracks and crevices into those hidden pockets that I was showing in one of the photos, those pockets of soil that were still holding on to moisture. And unfortunately, in some of our experimental translocation trials, we're finding more and more that we're getting quite a bit of mortality, and that's because our seedlings are just not getting their roots down quick enough. So in those trials, we were comparing both natural recruitment events across natural sites, but also in those translocates, so those artificial sites. The really interesting thing there is that our survival in our translocations was pretty much matching that in natural sites. So it's again linking into that paper I was talking about, where those significant recruitment events are really driven by those 1 in 30 and those 1 in 60 year rainfall events. But there's a big black box here. We need to start thinking about linking seed traits in with seedling and also plant traits. The challenge is that a lot of our plant functional traits are often based on adult plants. We're just starting to get a really good database for seeds, but for seedlings, Adrienne, you're agreeing. There's a big.
I'm nodding enthusiastically.
there's a massive gap of knowledge there, just because we're looking at ontogenetic shifts there, changes in relative growth rates, changes in root traits as well. So I'm really looking forward to this being the next space for us to dive into.
All right, thank you for that, Wolf. I'm going to pass it. You talked about this big reduction in seed size in Wallenbergia as a function or as a result of warmer conditions. Can you chat a little bit about, again, next steps? What do you think one could do if one wanted to intervene? Should we be thinking about translocating species in cases like this?
Well, I don't think translocation in this case can be applied because it's an Australian endemic species, so it's a variable for Australia, and it's the Alpine region in Australia occupies a very small portion of the continent. It's probably less than 1% of the continent, so it's very hard to find even sites in Australia where put these species. Alpine environments are currently not in extreme threat, but they will probably be if what we need to do is limit our footprint on the environment and try to contain this rising in temperatures.
Thanks, Rocco. All right, I'm going to move. There's been a couple questions for you, Lydia, and the ones that I think I might ask you to address are these ones that are asking about the difference between even and odd ploidy. So, for example, Chao Cunyo has asked, do you think there could be disadvantages caused by odd ploidy relative to even ploidy, and could parthenocarpy be a moderating factor?
It's a really good question. Looking at the different ploidy levels is definitely something we'd like to get a bit deeper into in next steps. With that particular Nacy example, we do have quite a few triploid species, which initially surprised us because they did produce seed, but some follow-on work that we did showed that a lot of that seed was produced apoptotically, and so basically, even though the seeds are kind of clones, and so whether that is, I guess, an advantage or a disadvantage when compared to the sexually reproducing species, we're not sure yet. I guess thinking about it from a seed biology perspective, if being a seed that's kind of a clone means you have a very synchronized germination among that population of seeds, maybe that's good if the right germination conditions happen to arise, but perhaps in a changing world, if those conditions are less likely to happen, then perhaps that's going to be more challenging for those populations of seeds. But I really am just guessing at this stage. It's something that we want to look into a lot more closely in next steps.
I found myself wondering if there was a simple database out there of ploidy that we could just look it up and then complicated species.
There is a genome size database that Kew maintain, but a lot of Australian native plants just haven't been looked at, or there's the odd publication here and there, but it definitely hasn't really been a focus in seed research, at least. Usually this sort of stuff comes up for phylogenetics or taxonomic studies. But yeah, there is a great resource out there that we can at least pull some data from. And I believe it's going to make its way into Austrates at some point if it's not already there.
Very good. I think that most of the questions that we've got have been addressed. Do any of my speakers have questions they'd like to ask of the other speakers? You have 1 minute. Lydia.
I was interested in the first question. I think it was from Jose about how we can use the temperature gradient plates to get beyond seed questions. And I wondered, Rocco, if you had anything to add to Wolf's answer.
No, sorry, I missed it.
I thought you were doing some great stuff looking at the next seedling stage.
I can take that one, Lydia.
Thank you.
we have actually tried to grow plants in the temperature gradient plate. That's what Lydia is referring to, and it does work. And I'd be happy to tell people about the trials and tribulations of doing that. But in the same way that you can put petri dishes in, you can actually put little tiny metal. It will also follow a similar temperature gradient. So it does have potential. And we are out of time. Nope, we're still going. Okay. Thanks, everyone. That brings us to the end of our session. We've got a short break, and then we're back for more talks.